Hi Bio 111 students, and welcome to week 7, chapter 6. We're going to be talking about enzymes today, and we're going to start off by talking about some characteristics of enzymes. So enzymes are highly specific. To substrate. So an example of that would be amylase, which is highly specific to starch. It begins to break down process of starches in your mouth in salivary amylase, and so there is an example of a highly specific enzyme that is highly specific to starch. Amylase actually doesn't work to break down anything else, so it's highly specific. Another characteristic is that they remain unchanged during the reaction that they're involved in. And what that means is that the same enzyme can go on to repeat the reaction again and again and again because it remains unchanged. Even though it binds temporarily to something, it remains unchanged and doesn't undergo changes that make it denatured or anything like that. It's ready to go and do the same job again right after. So another characteristic is enzymes are highly efficient. The rate that enzymes can work at can be 1 to 10,000 molecules produced. So 1 to 10,000 up to 10,000 to 10,000 products. And that's just in 1 minute. Another situation is actually that's just in 1 second. It's not even 1 minute. Another thing to remember about them is that they do not change the constant of equivalency constant. And that is that when reactions are moving one direction, they're also moving the other direction. So the enzymes actually, they might speed up the reaction, but it doesn't mean that the reaction is unequal going either direction. Especially in a reaction that we have an arrow that looks like this in the center where we have reactants and products, it doesn't affect that. It still remains constant. You can have allosteric control. Of enzyme action. And that is because a lot of enzymes require coenzymes and cofactors. So by providing those or withholding those, you can change the rate of enzyme action. Another thing that can happen to enzymes is that they can be allosterically inhibited. And You're probably looking at that word allosteric. What it means is that you can have shape changes of the enzymes, and if an enzyme's shape changes, it can no longer perform its job anymore. And so, by shape changing, by withholding the cofactors or coenzymes, or other ways, you can inhibit or change the rate of enzyme action. So that was characteristics. Now let's talk about how enzymes work. So you've probably seen a graph like this. A graph of a reaction. We have the reactants over here, and energy is needed for the reaction to go ahead, and this is called the activation energy. This difference here. Activation energy. Well, what enzymes do is they lower the activation energy. So instead of requiring this much energy, it might require this much energy. That makes it a lot easier for the reaction to proceed, and obviously it acts as a catalyst in that way, in that a catalyst is a thing that speeds up reactions, and this is exactly what's happening here. Activation energy is lowered, and so the enzyme is acting as a catalyst. So let's go through a few of these diagrams of product synthesis. Product synthesis is the making of a product by an enzyme. So let's first draw an enzyme. This is a model that you might see in your book. And so we have an enzyme here, and then we have a substrate. And so this is the substrate. This is the enzyme. And this area here is the active site. On the enzyme, and you can see that its shape is very specific to this substrate, because this substrate is shaped exactly like the shape that it's going to fit into. So the next part of the reaction, the substrate binds for a short time with the enzyme. So we'll have something like this. So this is called an enzyme substrate complex. The next step of the reaction, something happens to the substrate, and there's some slight changes in shape that happen as a result of being bound to our enzyme. And those changes could be as simple as this. So here's our enzyme substrate complex again, except now it has become the enzyme products complex. And the final step, what we're going to see, is the products leave. And our unchanged enzyme looks the exact same as this, and it is ready to encounter more substrate and repeat the whole process again. There is no change in the enzyme shape during product synthesis. Now let's talk about the enzyme activity rate. So, enzyme activity rate is affected by substrate concentration, and if we were to graph it, we would see this. Substrate concentration, we would label here. And we would label the enzyme activity rate over here. We're just putting EAR for short. And what happens is, as the substrate concentration increases, so does the enzyme activity rate, until we get to a point at which it flattens out completely. And that point is when 100% or close to 100% of enzymes are bound. So at that point, 100% of enzymes are bound, and so you no longer get an increase in the enzyme activity rate, because it's basically every enzyme that can grab a substrate has done it, and increasing the substrate concentration, well, it won't affect the rate anymore. Another thing that affects enzyme activity rate is temperature. And with low temperature, there is still some enzyme activity, because enzymes are not denatured by low temperatures. They are just slowed down. So what we see is, let's say this is our EAR, and this is temperature, and we'll make it degrees Celsius. So temperature in degrees Celsius. Let's say this is an enzyme that works inside the human body. So its optimum temperature will be 37 degrees Celsius or 98.6 Fahrenheit. And so let's say that is right around here. So what we'd see is a fairly low rate of enzyme activity, getting better as it approaches its optimal temperature, which is inside our human body, and then a pretty rapid decrease to 0. And the reason for that is because once we go much above the optimal temperature, you start to get denaturization of the enzymes, because they are proteins. And once a protein has been denatured, it is unable to do its job anymore. It's unable to, the shape is completely changed, and so it's unable to do anything that it was originally doing, can't bind to substrates, and so that really is the end for the enzyme. And so you just see this enzyme denatured at this point, and the activity rate plummets to 0. So we also see pH, which is low numbers acid, high numbers base, and let's say inside the human body an optimum pH would be 7.2 or 7.4, around then, right? Slightly basic. And so around that number, at that number, we're going to see optimal rates. So we're going to see the highest rate of activity, and around that number we're going to see drop-offs. These drop-offs are not the same as temperature, because it really doesn't like to be much less than 7.2 to 7.4, or much better or much higher than that. So really you're going to see a graph like this, unimodal, and you won't see much activity outside of this optimal zone. So another thing that changes enzyme activity rate are inhibitors. So one type of inhibitor is competitive inhibitors. Inhibitors. And so in our thing we had earlier, let's say we have an inhibitor that is like this, and so it can get in there and it can block. It competes for the active site, this area here, the active site on the enzyme, and then this thing, which is our ideal substrate, can't fit in there because this thing's in the way, and that's called competitive inhibition. And it competes with the substrate for the active site on the enzyme. So this is your substrate and this is your competitive inhibitor. This is the active site on the enzyme, and there's a competition between these two. And what it does is, initially, it slows the rate of enzyme activity. So here's, your optimal enzyme activity rate. That is only slowed by, all the enzyme being used up by substrate. And we will see a rate like this, where it's much lower at the start, but by the end of the time, it starts getting there to the point where it's it really basically, competitive inhibition, what it really does is it slows the initial rate, but it doesn't slow the end rate, the maximal rate as they call it. So now we have feedback inhibitors. Another thing that slows the enzyme activity rate. And feedback inhibitors, when you think about them logically, it makes a lot of sense. When your body is making a product, synthesizing a product, there gets to be a point at which you have enough made. And at that point, you really want the reaction rate, the activity rate, to slow or perhaps stop. So your body's maybe got enough protein of a certain type and has sent out the message, okay, stop. And this is how this happens. What happens is you have a reaction, the enzyme, and you have the substrate, and they go through, and they go through all of this. So then you get the enzyme substrate complex. Then you get the enzyme product complex, and it goes on and on and on. And finally, at the end, let's say we have another substrate, another enzyme, and the product looks like this. Well, that product, what happens is that product, when enough of it is there, comes back and makes contact with our original enzyme and changes it. So maybe now it's a bit more like this. So now our original substrate doesn't fit. And that's how feedback inhibition happens. Basically, an end product binds with your enzyme and causes an allosteric change. As I said before, it's a shape change, and that means that this is now inhibited from binding with the enzyme, this substrate. We also have a thing called allosteric inhibition, or allosteric regulators, and that is inhibition and activation. And again, allosteric, that relates to shape. Allosteric regulators. And it's called regulators because it changes the rate of the enzyme activity. Rate of enzyme activity, or the enzyme activity rate, is changed by these regulators. So first we're going to talk about, on one side, let's do it in red. So, inhibitors. And we're doing it on red because it basically is like a stop sign. So, we have our allosteric inhibitor here. We have a substrate here. And much like our original feedback inhibition, where this would be product, in this case it's not a product, this is just, it does the same thing as what the product did before. It changes the shape of when it engages, when it binds to the enzyme. It changes the shape of the enzyme, and so it can no longer bind. So it's stuck out here. That's an inhibitor. Now we're going to use green for activators. Green light, go. So this is an activator, and we have the same model, except this time, let's say our it looks like this, our enzyme, and again this is our substrate, and it can't bind because the shape is wrong. So what the activator does is when it binds to the enzyme, we get a shape change, much like before, but this shape change is favorable for the enzyme, and so the enzyme can bind. Now, just to get you give you something, a real life example of this. This might be penicillin, and this could be a bacterial enzyme. So an enzyme in a bacteria And what the penicillin, PCN, is doing is, when it binds to the bacteria, it changes the shape of the enzyme and is an allosteric inhibitor, because now this enzyme can't do its job. And that's all I really have time for today. That's all you need to know about enzymes for chapter 6. And I'll talk to you tomorrow about energy use in the cell. Thank you. Bye.